Okay, so I'd like to start by thanking the organizers for inviting me to speak here. It's really a privilege, and also the organizers of the program for inviting me to participate in the program. It's been very enjoyable over the past few months. I'm going to talk to you about a more applied problem than a lot of the talks we've had so far, but hopefully it'll still be of interest. And the work I'm going to talk to you about is joint work with Charlie Elliott and Björn Stinner, who are both at Warwick. So what I'm interested in doing today is modeling processes like the one you see on the slide here. So this is just the directed migration of cells. On the left, you see chemotaxis of dictyostelium. So these cells, they're migrating up a chemical gradient, and there's a source of chemoattractant to the right of screen. So that's why all these cells moved rightwards. On the right, you saw persistent migration of fish epithelial keratocytes. So these are cells which live in the scales of fish skin, and they migrate around the surface of the fish looking for scratches in the scales which they proceed to heal. So for them, it proves optimal to migrate in a different fashion, so they migrate with a fixed shape, in a fixed direction, and with a roughly fixed speed. And we'd like to try and develop a modeling framework in which we can simulate these different types of cell motility. This is important because cell motility plays a fundamentally important role in many biological phenomena. For example, wound healing, immune responses, and the metastases of cancers are all largely governed by the ability of cells to migrate in a directed fashion. So my sort of naive understanding of cell motility is that it can be split into 4 stages. You have an initial stage where the cell polarizes and develops a front and a back, and here it determines its direction of motion. So it's going to move forwards. Then, as we heard in a nice talk last week, you have protrusion. So actin polymerizes at the leading edge of the cell, and this generates protrusive forces. This protrusion at the front of the cell forms adhesions with the substrate, and then the rear de-adheres from the substrate, and contractile forces are generated by proteins such as myosin motors, which pull the rear behind the protruding front. So that's a schematic for how eukaryotic cells, which are cells in animals, for how they move. So in the research I want to talk to you about today, our goal was to try and develop a robust modeling and computational framework for investigating cell motility. And within this framework, we proposed that we could model the mechanics with a geometric evolution law for the motion of the cell membrane, and we modeled the biochemistry with a PDE posed on the cell membrane. Now, the advantage of this modeling setup is a reduced dimension. Effectively, if we want to study cell motility in 3 dimensions, we have a PDE on a 2-dimensional surface and a PDE for a 2-dimensional surface. And that proves useful when you want to simulate these models. This is a contentious way to model cell motility if you talk to biologists, because there are bulk phenomena which you neglect with this modeling process. But the only thing to say in response to that is there are lots of very applied studies that use exactly this modeling framework. So I think it is of some relevance even to the more applied and experimental people. Once we have our models, which will be a nonlinear surface PDE and a geometric evolution law, we'll discretize them with a numerical method based on surface finite elements, and I'll go into that in a little bit of detail. So an outline of the talk is that I'll start with the modeling. I'll try and highlight the assumptions that go into the different models. Then I'll present the discretization, and in particular, I'll focus on some simplified problems for which we can do some analysis. I'll present some examples of how we can model real-life examples of cell motility, and then I'll discuss some more recent work on parameter identification. So here we've tried to develop a tool that is of use to biologists, such that we can identify parameters in the model equations such that our simulated cells best resemble experimentally observed cells. So in the modeling, well, as I told you, we have a PDE posed on the cell surface that models polarization. And the assumption here really is that the reactions that take place that lead to polarization and gradient sensing, they occur either in the cortex close to the cell membrane or on the cell membrane itself. And this is really motivated by pictures like the one you see there. So that's a dictyostelium cell migrating in a flow chamber, and it's been transfected with a fluorescent protein that makes actin in the cell fluoresce green. And what one observes is that filamentous actin, which is correlated with protrusion of the cell, stays localized on or near the cell surface. So if we assume that these reactions can be modeled as taking place on the surface of the cell, then one way we could model this process is to propose a surface reaction diffusion advection system posed on the evolving cell membrane. Okay, so this is going to be the model that we adopt for the biochemistry. And I'm working on an evolving surface, so I have to consider the material derivative rather than the usual time derivative with respect to the material velocity of the surface V. And I also replace the Cartesian differential operators with surface, so for example, the Cartesian gradient with the surface gradient and the Cartesian Laplacian with the Laplace-Beltrami operator. I'm interested in a reaction diffusion system where the species diffuse independently of each other and they're coupled through this nonlinear reaction G. So this is our model for the biochemistry. For the motion, we propose a geometric evolution law. And the idea here is that we assume a force balance on the cell membrane. And for the purposes of this talk, I'm going to assume that all of these forces act in the direction normal to the cell membrane. This isn't necessary, but if you talk to experimentalists, they say that they believe material points on the cell surface move in the normal direction only. So the forces we take into account are the resistance of the cell membrane to stretching. A very simple model for this is to consider a spatially uniform surface energy density. And the force contribution that this will lead to is a mean curvature flow type force. So that's the first line on the slide here. Throughout, H is going to denote the mean curvature of the surface, by which I just mean the sum of the principal curvatures. The cell membrane resists bending. A simple model for this is to consider a spatially uniform bending energy. And this leads to, and we can model the bending energy with, say, the Willmore functional. And this leads to a Willmore flow type force. We also have a viscous force that opposes the motion, so this is just going to be proportional to the velocity. And finally, we have this coupling between the evolution of the surface and these PDEs posed on the surface, and this is through this protrusive force that depends on the surface field. You also observe that when these cells migrate, their surface area may change a lot, but the volume they enclose stays roughly constant. So we model this through including this spatially uniform force lambda, and you can think of lambda as an interior pressure that enforces volume conservation. So it's a Lagrange multiplier such that the volume enclosed by the surface remains conserved. And because I assumed a force balance on the cell membrane, bringing together these force contributions gives me my evolution law, which looks like this. So it's an equation for the normal velocity of the cell. The simplest form of coupling to the surface fields is to assume we have protrusion or retraction proportional to these quantities that live on the surface. So the sign of the ith component of this vector Kp will tell you whether the aith species promotes protrusion or retraction of the cell membrane. I've also included these Fx, the external forces, and I'll show you how through those forces we can include sort of toy models for more complicated phenomena. So interactions with other cells, adhesion to a substrate, or migration in the presence of obstacles. This is a nonlinear fourth order geometric evolution equation because the Laplace-Beltrami of the mean curvature enters, and the mean curvature itself is a second order operator. So our numerical method, I'm going to present in a second, has to try and approximate a solution to this evolution law, and also the PDE that's posed on the cell surface. So for the numerics, I want to sort of present a simplified problem, partly because I think it makes the ideas of the discretization clearer, but also partly because this is in the setting in which we can do some analysis. So let me just for this example, let's go to a world where the surface is known. It's no longer an unknown. I know my surface. It's smooth, and it's moving with a smooth velocity v. So v is now a given function, and it's the velocity of my known surface. And let me consider the linear parabolic equation on this evolving surface. So the correct form of that equation is the one you see here. And if the boundary of the surface is non-empty, I augment this with zero flux boundary conditions and suitable initial data. Then, if you test with a sufficiently smooth test function, integrate by parts, you end up with a weak formulation, which looks like this. And Zhuk and Elliott, in their paper in 2007, they showed this Leibniz formula for the time derivative of the L2 inner product on the surface. And if I apply that in a weak formulation, I get this version of the weak formulation in the bottom line. And that proves to be the convenient one with which to formulate a numerical method for this problem. So I'm going to now illustrate a numerical method to approximate the solution to this weak formulation that you see here, and it's going to be the evolving surface finite element method developed by Zhuch and Elliott.
So this velocity, this tangential velocity which you are looking there, that plays a role?
V has both normal and tangential components, yeah? But what do you mean, does it play a role, sir?
I mean, how did you model it?
So for this particular example, it's prescribed.
Yeah.
But I'll get on to how we actually deal with it when it's an unknown. So the idea for the numerical method here is that so this idea dates back to the work of Zhuch in the late 80s and the early 90s. And the idea is that you take your continuous surface and you approximate it with a triangulated surface. So now my triangulated surface gamma h is the union of triangles such that the vertices of each triangle lie on the continuous surface at all times. So the triangulated surface interpolates at the vertices the continuous surface. Then I can define a finite element space over my triangulated surface as the space of functions that are continuous over gamma h and such that the functions on each triangle are linear. A basis for this space is given by the set of piecewise linear nodal basis functions. So these are functions in the finite element space that are 1 at a single vertex and 0 at all others. Then, okay, so my assumption here is that the vertices of my triangulated surface lie on the continuous surface at all times. So one way to ensure this is to move the vertices of the triangulated surface with the material velocity of the continuous surface. If I do this, this induces a material velocity of my triangulated surface, which looks like that first line there. And then I can write down the material derivative of a function on the triangulated surface with respect to this material velocity. And Zhuch and Elliott in their 2007 paper showed this remarkable property that the finite element basis functions, their material derivative with respect to the material velocity of the triangulated surface is 0. And this proves particularly useful in the numerical method. The surface gradient on the triangulated surface should be understood element-wise, and it can be written in that way, where nu h is the normal to the triangulated surface. So then the numerical method to approximate the solution to the surface heat equation now looks like this. All you do is you take your integrals over the triangulated surface, and your trial and test functions now live in the finite element space. And then when we observe that the basis function satisfied this transport property, the first term on the right-hand side of that equation is 0. So the top equation, the first term on the right-hand side of that is 0 if I insert the basis functions there. So this finite element formulation is equivalent to a system of ordinary differential equations for a coefficient vector u, where M is just a mass matrix, and now it's but now it's time dependent because of course the domain of integration is time dependent and the basis functions are time dependent. A is a stiffness matrix, which is again time dependent, and F is the discretization of the right hand side. So this is essentially the evolving surface finite element method for this linear parabolic equation I just showed you. And it's possible to do quite a lot of error analysis for this numerical method. So the idea behind the error analysis is that we lift the discrete solution from the triangulated surface to the continuous surface such that we can compare the discrete solution and the continuous solution. And in their original paper where they formulated the method, Zhuch and Elliott showed optimal energy norm error bounds in the semi-discrete case. So this is continuous in time, discrete in space. They showed optimal L2 error bounds in a later paper. And they showed an optimal error bound for a full discretization, where we discretize in time with an implicit Euler method. It's also interesting to look at other time discretizations of the system of ODEs because the mass matrix and stiffness matrix are time dependent. And in particular, Zhuch, Lubitsch, and Mansour looked at higher order Runge-Kutta methods for this system of ordinary differential equations. And together with Lubitsch and Mansour, I looked at BDF, time stepping schemes. And again, you can prove optimal error bounds for numerical methods. There's an observation which will come up in the method I'm going to present to you today, and that's that I said I'd move the vertices with the material velocity of gamma. And there's no reason that moving the vertices with that velocity gives me a good triangulation. So an idea to alleviate this is to move the vertices with this velocity v sub a, and Elliott and Stiles used this in a paper in 2012, where v sub a is the material velocity plus some arbitrary tangential velocity field. This is going to give you a new numerical method, which I'll call the ALE, evolving surface finite element method. And if I move my vertices with this velocity, then the triangulated surface moves with a different material velocity, the one that's given there. The material derivative on the triangulated surface is now taken with respect to this new velocity field, v sub a. And now the basis function satisfies this new transport property, that the material derivative with respect to this velocity v sub ah is 0. And we want to try and exploit this transport property in a numerical scheme. So I take the weak formulation I wrote down earlier, and then I note that if I take the material derivative with respect to the material velocity, I can rewrite it in terms of the material derivative with respect to this material velocity plus the arbitrary tangential velocity, so this v sub a, plus this extra term, which depends on the tangential velocity. And if I plug this replacement into the weak formulation, I get this new weak formulation below. And it proves convenient to think of this as the weak formulation one discretizes with the ALE surface finite element method, because you can use this transport property of the basis function.
Then the optimization starts, and after some iteration,
and now the ALE evolving surface finite element method just reads almost exactly like before. This line here, where the integrals are over the triangulated surface, and our trial and test functions live in the finite element space. And then noting this transport property of the basis functions, we see that the first term on the right-hand side of that finite element formulation is 0 if I put the basis functions into it. So the finite element scheme is equivalent to a system of ordinary differential equations for a coefficient vector u. And all I've picked up that's different from the Lagrangian case is this extra matrix B. And B is a non-symmetric matrix with coefficients given by the last line there. It's also possible to do the error analysis for this numerical method. And Charlie and myself finished the paper on that during my stay at Cambridge. So for implicit Euler or BDF two-term discretizations of this numerical scheme, we can show stability in fully discrete optimal error. Okay, so this was a world in which we could prove something, but for the actual problem I'm interested in talking to you about today, the surface is of course an unknown. So we don't have a prescribed velocity. And it turns out that because we have to solve a geometric evolution law, we have to work a little bit harder in the way that we approximate the surface. So we actually use isoparametric quadratic surface finite elements. And the idea here is that, again, gamma is approximated by a triangulated surface, who's made up of the union of triangles. But now each of the triangles is the image of a linear reference element under a quadratic map. Okay? And this idea was used, for example, by Heine, I think, in 2003 and 2004. And then one can define a finite element space over the triangulated surface by taking the space of functions that are continuous over gamma h, such that the functions composed with the map are quadratic on each triangle. Okay, and I'll try and make clear why using these isoparametric quadratic surface finite elements is a good idea for this problem. So let me give you an idea of how we solve the geometric evolution equation. And again, let me illustrate the principles of the discretization in a simplified setting. So let's think for a second that we just consider mean curvature flux. So that says that the velocity is minus the mean curvature vector. And let's consider the approximation with the algorithm proposed by Zhuk in 1990. Essentially, the algorithm Zhuk proposed in 1990 underpins the principles of the discretization for the more complicated evolution law that I'm looking at today. So Zhuk's scheme is based on this identity here, which says that the mean curvature vector is just minus the Laplace-Beltrami of the identity map. And then the scheme essentially just says the spatial discretization step is to replace the continuous surface with a triangulated surface. replace the identity map with an approximation of the finite element space.
and different targets.
And then for the time discretization, since the geometry is unknown,
More important
so at time m, we do not know the geometry at time m plus 1, it proves convenient to discretize in time explicitly with respect to the geometry.
case you have
So the numerical method to approximate the solution to mean curvature flow looks like this here.
we solved it like
And then when we compute xhm plus 1, we set that to be the new identity map on gamma hm plus 1.
find the optimal solution, perturb it by
Essentially, that's exactly the same idea for a more complicated evolution law.
as a constraint.
But now we work a little bit harder since this is a fourth order equation, and I want to discretize it with C0 finite elements. So I use a second order operator splitting technique. And in fact, the numerical scheme we use, which is based on this weak formulation here, is a scheme proposed by Barrett, Garka, and Nuremberg. And their scheme is particularly nice for this application because they observe the fact that this evolution law gives me an equation for the normal velocity of my surface. I can do anything I want in the tangential direction, and I still satisfy the evolution law. And they make use of this fact, and their scheme induces a tangential velocity, which has nothing to do with the material velocity of the surface, but in practice it gives you a very good mesh. So that's the role of this tangential velocity, to give you a good triangulation. I also have this constraint, this Lagrange multiplier lambda, which I need to determine to enforce volume conservation. And there's a very nice method laid down for doing this that was proposed by Benito, Nochetto, and Paoletti, which is the one we adopt, where you basically employ a Newton iteration to determine the Lagrange multiplier.
So at the end, again,
The choice of this isoparametric quadratic surface finite element space is motivated by results such as this one here. So essentially, the difficult term in the evolution law to approximate is this surface gradient of the normal, or the Weingarten map. And Klaus Heine showed that if you use these isoparametric quadratic surface finite elements, and you considered a scenario where the surface was given to you, then you can construct approximations of the Weingarten map that are order h in the L2 norm. I'm not aware of any results that show convergence of an approximation to the Weingarten map with linear finite elements. Triangulated surfaces. So that's the motivation behind using these quadratic surface finite elements. So that's essentially how we solve the evolution law. We still have to solve the surface reaction diffusion system. But basically we just use the ALE SFAM I presented to you earlier for the linear parabolic equation. We have to use this ALE evolving surface finite element method because the scheme of Barrett, Garka, and Nuremberg generated this tangential velocity, which was independent of the material velocity of the surface, which is only in the normal direction. And we get a time step restriction anyway from solving the evolution law, which means we can treat the reaction kinetics explicitly in this discretization. Okay.
Yeah, you're absolutely right.
So that's essentially a numerical method. We approximate the surface reaction diffusion system with an ALE evolving surface finite element method. And we approximate the evolution law with a parametric finite element method based on the method of Barrett, Garka, and Nuremberg. The big advantage of this approach over other standard approaches is one of reduced dimension. We solve the surface PDE and the surface evolution law with triangulated surfaces. So for a d-dimensional surface, our algorithm is d-dimensional. Other standard methods to approach this problem would be, say, level set or phase field methods. There you'd approximate your PDEs with PDEs posed in the embedding space, and so your algorithms would become d plus 1 dimensional. And for the kind of problems we want to consider, which are these computationally intensive parameter identification problems, the efficiency of this numerical method, I think, will prove crucial. There are certain issues. For example, if you think the physics in the interior of the cell are important, then it's not, well, it's extra work to include bulk phenomena within this setup, because we only have a triangulation of the surface, and you somehow have to marry this to a triangulation of the bulk. And if you're interested in topological chains, then it probably makes sense to consider a completely different framework, as we work with a parameterization with a fixed topology.
You do not know in advance where your switching points for the SWOT coverage are. So you do not know where.
So I wanted to present a video which just illustrated the benefits of using this tangential redistribution scheme. So on the left, I'm going to solve this surface evolution law surface PDE system, and I'm just going to move the vertices with the material velocity, so in the normal direction only. On the right, I'm going to use this scheme proposed by Barrett, Garka, and Nuremberg, which induces this tangential velocity that gives you good mesh properties. And what you see is that on the left, the surface rapidly becomes unsuitable for computation, whereas on the right, this redistribution of the vertices gives you a very nice mesh.
Yeah,
So that's what makes this scheme particularly robust for these types of applications.
are zeros in this case. It was an artificial.
Illustrate some examples of what we can model by considering some real-life examples of cell motility.
Were not in code, it was
asymmetry
the memory
generalized.
of
Sorry?
the computer we use. It was a special computer with 0.
Okay, so there's a PDE on the surface here, which is a Turing-type reaction diffusion system, and the initial conditions are a symmetry break, which generates.
we are able to handle.
But this was just an example cooked up to show why this tangential redistribution is useful. So let me give you an example of a real-life example of cell motility. We considered modeling the first video I showed you on the very first slide. So this is so-called pseudopod-driven chemotaxis, and it's essentially where a cell migrates up a chemical gradient.
It was a very funny situation.
There's a very nice model for this process proposed by Meinhardt, where he considered a 3-component reaction diffusion system, which you can reduce to a 2-component system of the form I'm showing you here. You have a local activator A, a local inhibitor C, and essentially you have a chemotactic signal, which is this term S. S feeds multiplicatively into the reaction kinetics of the activator, and the natural dynamics of this activator A will amplify this chemotactic signal. So this gives you a mechanism for amplifying very weak chemotactic signals. If the signal increases a little, A will naturally increase a lot. We model the signal with an Ornstein-Uhlenbeck mean-reverting stochastic process, and this comes from a simple model of receptor-ligand dynamics. Okay. And so now, where the signal is elevated a little, we expect A to become elevated a lot in that region. And so that's the direction in which we want our cell to move. So it's natural to couple protrusion of the cell membrane to the surface density of A. So that's what we do in the evolution. So you can run simulations with this model. For example, here we have a signal whose form is given by this function mu. So we have a source of chemoattractant located at the point xc.
We have no problems with the Maratov effect. I don't know why, but we have no problems with it. Maybe this is the reason, but this Maratov effect is also appearing, I think, in other things.
Is that any better?
It is better.
I can try this
say
as
it's
well.
perfect.
Let me leave this one.
Wait, they're just tracks.
Well, then you'll have to take my word for it that as you increase the strength of this
If you take that stick, you can go actually just point
True.
the curves to people so that.
Yeah.
Yeah.
It's a good idea. We have a signal which looks like this, and we have a source of chemoattractant that's somewhere to the left in each of these images. Okay, and you can think of this parameter rho as being proportional to the gradient of the signal here. So when we have no signal, the cells migrate in a random direction. These paths here are the trajectories of centroids of these cells. So it's the center of mass of each cell. As we increase the strength of the signal, the cells all migrate up the chemotactic gradient. So each of these paths goes towards the left, which is the direction of the signal. And you can try and quantify whether or not your cells resemble experimental cells. And I don't want to go into all the details here, but there's good agreement with what you measure in experiments. So you can look at lots of different measurements that are commonly used measures of chemotaxis, and the simulated cells seem to match up very well with experimental cells. And this is without any particular parameter fitting. We just use the parameters from Meinhardt's original models, and for all the other parameters, picked physically reasonable values. You can also look at toy models for more complicated scenarios. So, for example, we saw this video here. This is a neutrophil chasing a bacterium in a sea of obstacles, and it pushes these obstacles out the way until it eventually engulfs the bacterium. And on the right, you see a bunch of dictyostelium cells chemotaxing together, and it appears that when they get in close proximity, they adhere with each other. So we thought about ways in which one might model these phenomena, and the idea is to include extra potentials in the model to account in a phenomenological way for these phenomena. So for example, for the obstacles, we could think the obstacles move according to a viscous evolution law and that they're rigid. And then I could posit the interaction between the cells and the obstacle just through this repulsive potential, which becomes prohibitive if the cell membrane tries to intersect the obstacles and decays to zero as soon as the cell membrane moves a small distance away from the obstacles. Then you get a force contribution in the evolution law, which you can calculate, and it'll look like this. And you get an opposite force contribution on the obstacles, and it's sufficient to just model that at the center of the obstacles because I've assumed they're rigid. So this is the force on the obstacles in the viscous evolution law. If, rather than modeling repulsion due to obstacles, you wanted to model adhesion with other cells, then you could replace the repulsive potential with a Leonard-Jones potential. This is adhesive at small distances and becomes repulsive at very short distances. So it'll promote adhesion, but when you become too close, it'll prevent self-intersection. And when you simulate these types of things, looks a lot better on my screen than it does on here, but anyway. You get a cell migrating through the presence of obstacles and pushing them out of the way. So these are just toy simulations, but I think it illustrates what one can do within this modeling and numerical framework. And then on the right, I have all of these cells which are chasing the source of chemoattractant, and between them is a Leonard-Jones potential. So when they get close together, they want to adhere to each other. And eventually, one of them wins the race and catches the source of chemoattractant, but you also see that they adhere when they're close together, but they never self-intersect. Okay, so within this framework, you can look at these types of scenarios. I think that was the point of this slide. But now I want to talk to you about some more recent work where we've been trying to develop a tool which may actually be useful to biologists. And so the point here is that can we develop a methodology for fitting model parameters to experimental data? And in particular, can we use this sort of experimental imaging data that's becoming ubiquitous in biology to quantify things within the models? So a lot of the parameters are just picked out of thin air because they're phenomenological, but perhaps we could use the experimental data to quantify the relationship between protrusion and these surface fields, to get some estimate on reaction rates of reactions taking place on the membrane, or even to estimate material parameters. So what one sees is that in experimental studies, it's pretty hard to measure things like surface tension of a cell membrane, and depending on the experimental setup you use, you can get lots of different measurements. So maybe this would be a tool that would be useful to experimentalists in estimating these sorts of parameters. And so mathematically, the problem can be formulated in this way. I have a set of parameters C, which I want to identify. They could be diffusivities, reaction rates, surface tension of any rigidity, for example. And I have a series of observations. So at ns distinct times, I have an observation of a surface and associated with it a concentration. Okay, and these are the observations I want to fit to. Then I'm going to define by gamma and A the solution to my model equations, and this depends on the parameters I want to identify. And then the identification problem is to identify parameters such that the solution to my model equations is as close as possible to the solution to the observed data. And so the first thing you need to define is what does it mean for the solution to the model equations to be close to the observed data. And the first idea we had for doing this was through this objective functional here. And basically, I'm going to call this the sharp interface objective functional. This component of it measures the difference in position between the observed data and our computed curves. And the idea is that you take the distance function to the other surface and integrate that over each surface. And so this first part here gives you a measure of the difference in position of the two curves. We also need to measure the difference in concentration between the two curves. And to do this, we just evaluate the difference in concentration with the closest point on the other curve or surface. So this second part of the objective functional gives you a measure of the difference in concentration between the two surfaces. The issue with this formulation is one of smoothness. So this is non-smooth, and to see that is easy, just consider the second part of the objective functional. The closest point on the other surface may jump, or it may not be uniquely defined. And the optimization method we're going to use to try and identify the parameters will assume that the objective functional depends smoothly on the parameters. So we decided to try and think of a different way of formulating the objective functional, and the idea is, why not approximate our sharp interfaces with diffuse interfaces? So if you give me a curve, I can define this phi epsilon, where epsilon is a positive parameter which governs the width of my diffuse interface, and that'll give me a diffuse interface representation of the curve. So it takes the surface and it thickens it into a diffuse interface. And similarly, I can define an extension of my concentrations away from the surface through this A epsilon. So A epsilon here is just extends the concentrations constantly in the normal direction and weights them by the distance from the curve. And this gives me a diffuse interface representation of the position and the surface concentration. So to give you an idea of what I mean, if these are the three curves I get from a simulation, then these would be the corresponding diffuse interfaces, the corresponding diffuse interface representation of the concentration on these curves. But now I have an alternative way of formulating my objective functional, and that's just to take these diffuse interface representations of the curve and the concentrations, and I could define them on the same bulk domain omega, and simply take their difference in L2. And this is another way of defining the difference between my computed curves and the data I want to fit to. The catch with this approach is that at every time I want to compute the objective functional, I need to initialize a diffuse interface representation of my observed data and of the curves I compute. So this is much more expensive computationally. I can still do the computations with the sharp interface method I showed earlier, but at each point that I need to compute the objective, I need to initialize these diffuse interfaces.
And you do that over the interval 0 t?
No, so I only have the data, sorry, I should have been clearer. I only have the data at a series of distinct times. So I goes from 1 to ns. The data would correspond to pictures, snapshots you get from experiments. And so those are I want to find an evolution of the surface and the concentration such that at these distinct times, I fit the data as well as possible.
But still, I mean, you run the initial, what I mean is you run the initial value problem for the whole time.
I solved the initial value problem over some time interval, yes. And at a series of distinct points within that time interval, I compare with these observations. Yep.
It might be an error which accumulates, right?
Yeah, that is true.
I don't know how stable it is.
So that's true, but that's kind of the point of these weights here. So you would weight each of these contributions to the objective functional, and as you say, you might be much more confident of your observations closer in time to your initial data. So the weights for I equals 1 might be much larger than the weight at I equals ns. For example, that might be one way of doing it. This is an initial approach, so this is quite recent work. I think there may be ways to do this better. Okay, so now the minimization problem is easy to formulate. I have an objective functional, and I just want to minimize over parameters in some admissible set the objective functional, which is either given by the sharp or diffuse interface formulation. It's natural to constrain the parameters to lie in some admissible set, because if I have diffusivities, for example, they should be non-negative, or surface tension should be non-negative, and so on. Okay, so now I need a method of trying to approximate the solution to this minimization problem. And what you observe, if you write it in the correct way, is that the objective J can be written as the sum of squares. So the minimization problem is just a nonlinear least squares problem. And as this was a first go at these types of optimization problems, we decided to use a method that was robust for these nonlinear least squares problems. So we used this Levenberg-Marquardt algorithm, which is meant to be robust for nonlinear least squares. The idea is you consider a linearization of these chi's. This is valid for smooth chi. And then you compute so the algorithm is iterative. If you give me an initial guess for the parameters, I compute an update according to this formula here. So if you took mu to be 0, for example, this is the Hessian of the linearized objective functional. This is minus the gradient. So computing an update according to this formula is just Gauss-Newton's method. If you take mu to be large, this is minus the gradient, so this is steepest descent. Choosing mu adaptively is what gives this method its robustness. So you have mu, which is some damping parameter, which is strictly bigger than 0, and makes this method quite robust. And we choose mu in a standard way. There's lots of literature on that. We don't have access to the Jacobian of chi analytically, so we have to evaluate it with finite differences. So we have to solve a model equation forwards in time multiple times for each of the components of this Jacobian, but these can all be done in parallel. They're all independent of each other. The stopping criteria for the algorithm would be if the objective was below a certain tolerance, if the gradient of the linearized objective was below a certain tolerance, this would suggest you're in a local minima, or if the algorithm stagnates or you reach a maximum number of iterations. And what was surprising to us was that the method was very robust, even though if I take the sharp interface formulation of the objective, this chi is not differentiable. But even though that was the case, for both the sharp interface and diffuse interface formulations of the objective functional, the method seemed to perform pretty well. Just to illustrate that with some examples, we looked at a numerical example. So we generated some artificial data by solving a model for keratocyte motility forwards in time. So the model is here, and the parameters we want to identify are the protrusive forcing strength, so the coupling between this surface density A2 and the surface evolution law, the reaction rate in the reaction diffusion system, and the bending rigidity of the surface. So generating some data with these true parameter values and picking initial guesses that were reasonably far away from the true values, so that the solution qualitatively at these initial guesses is very different to the true solution. The algorithm spits out estimates of the parameters that are quite close to the true parameter values. So relative errors that are very small. In biology, inherently data is noisy, so we also had a look at what happens when you perturb the target data by noise. So we perturb the observations by normally distributed noise of varying standard deviations, and estimates of certain parameters appear robust. So what's in this table here is the mean and standard deviation of the relative error of the estimate of each of these parameters over 100 runs with noise of varying standard deviation. And what you see is that estimates of certain parameters like the reaction rate seem very robust to the presence of noise with one formulation of the objective, but other parameters like the bending rigidity show no robustness at all. So this needs some more thought, I think, but this was an initial test in this direction. We also looked at an application where we apply this methodology to experimental data. So we obtained some data from experimentalists at Warwick, and basically they gave us data on fish and yeast. So fish and yeast is somehow the perfect cell with which to start looking at these problems. It's ellipsoidal, and it just elongates along one axis. So the geometry is very easy to fit to. You can think of a cigar shape that's getting longer. And these fish and yeast cells can either grow from a single tip or they can grow from both tips. But we picked a strain of fish and yeast that only grows from one tip. Growth of this cell is correlated with a cell-resident protein called CDC42. So that's, in this grayscale image, that corresponds to black. And basically what you should see is that only tips where CDC42 is elevated grow. And that's exactly what happens to this cell. This plot here shows CDC42 concentration along the cell membrane, and we see it stays localized at a single point in the cell membrane. So this is the data we want to test our identification algorithm with. The model now, okay, so the data comes to you as a grayscale image, and it's segmented using some software developed by systems biologists at Warwick to give data of the format we want, so a surface and associated concentrations. The model now is just a single species equation on the cell surface for the species CDC42. And it's reasonable in this case to think of CDC42 as satisfying a surface heat equation. So on the timescales we're interested, reaction kinetics don't play a significant role. But we still have coupling with the evolution law, and the coupling is through this function g. So basically when experimentalists look at this model, when experimentalists looked at these fish and yeast cells, what they observe is that fish and yeast grows if the CDC42 intensity is above a certain threshold. Otherwise, it doesn't grow at all. So there's sort of this threshold dependence on CDC42 concentration. And to approximate that with something smooth for the numerical method, we regularize this threshold function. So you have no growth if the CDC42 intensity is below a certain level. If it's above a certain level, you grow with a maximal rate. And Kreg is a small regularization parameter that smooths out this threshold function. So the parameters we want to identify are the threshold, K1, at which growth starts, and the maximal growth rate, K2. So we have two parameters to identify. And to show you some results of what you get out from these types of simulations, so the dots on these images are experimental data shaded by CDC42 intensity. The curves are the solutions with our identified parameters. And I've only shown you the tip that's growing because the other tip is boring, it's stationary. And so you see that there's reasonably good fit between what you get with the identified parameters and what you see in experiments. And also the two different formulations of the objective functional give you qualitatively similar results. And you can also look at the actual parameters that you identify. So the two different formulations of the objective functional, the left is the sharp interface objective, the right is the diffuse interface one, essentially give you very similar estimates of each of these parameters. So these, on the x-axis here are different cells, and on the y-axis is this parameter corresponding to the growth rate, and here on the y-axis is this threshold level. And what you see is that the parameter estimates with the two different objective functionals are essentially the same in a lot of cases. And also, this parameter here is cell-specific. It can't be compared across the population. It depends on how much of a fluorescent protein the cell produces. But this parameter here can be compared across the population of cells. And so in an experimental study, these authors here constructed a crude estimate of the maximal growth rate of the tip, and they concluded it was approximately 0.013, but there's quite a lot of deviation through the population of this value. And what we see is that we have growth rates that are pretty much in the same region. So there's good agreement with experimental data as well. So that's pretty much most of what I wanted to say. To sum up, we presented a general model for cell motility based on an evolution law for the mechanics and a reaction diffusion system that models the polarization and the biochemistry. And the modeling approach that we adopted is motivated by efficiency thanks to the reduced dimension and it's a model to resist. We presented a robust and efficient numerical method for the simulation of the model equations, which is based on moving triangulated surfaces and surface finite elements. And we can illustrate the effectivity of this approach by reproducing real-life examples of cell motility. I also presented an algorithm for parameter identification in these types of models, and we attempted to quantify a growth model for fish and yeast. And the result, the parameters identified were similar based on two different formulations of the objective. In terms of where we wanted to go, well, I told you in the first slide that this all happens on a substrate usually, and I've sort of neglected the substrate since then. So we have some ideas for preliminary models of how one might include it. So I still have a surface reaction diffusion system which generates a protrusion, but now the protrusion adheres to the substrate, and this happens through the dynamics of binder proteins which live on the cell membrane. And these binder proteins, based on a model produced by some biophysicists, they satisfy a Kahn-Hilliard type evolution law on the surface. So these binders phase separate, and now the strength of adhesion to the substrate depends on the surface density of these binders. They're naturally attracted into the protrusion, and they force the protrusion to adhere to the substrate. It's in early stages, this model, I think, but I think there are some ideas for that direction. I think we also need to account for bulk phenomena. We need to be able to include physics that happen in the interior of the cell. And as you're probably aware, there's lots of scope for analysis of these types of problems. It would be nice to establish an existence theory for simplifications of these problems, and it would be nice to try and attempt numerical analysis for something closer to the full model. There are also lots of little sub-problems. For example, looking at these simulations, you see that surface tension plays a role in determining whether self-intersections form or not. So if I switch off surface tension and I just have Willmore flow with forcing, then you very quickly develop self-intersections, and it might be interesting to try and quantify how forcing, surface tension, and the development of self-intersections are related in a simplified model. So that's everything I wanted to say, and I'd like to thank you for your attention.
Are there any questions or remarks?
For this inverse problems where you had these very good results, when you created this artificial data, did you use the same algorithm, or?
The artificial data, yeah, is just solving the model equations forwards in time.
An inverse crime.
Possibly, we have it, yeah.
So you don't have any, because you don't have any additional regularization in your
No,
function.
but this Leeuwenberg Marquardt algorithm has implicit in it
Yeah,
has some
of course.
regularization,
Yeah, with
doesn't it?
discretization, but
Yeah.
so.
Yeah. But yeah, that's something to look at.
Maybe I will have a question. In these kind of problems, do you have any fluid structure interaction or no?
Not in this one, but I think in real life, yes, and so that should be included. I mean, the way we've modeled it, not at all, but in a more general model, it should be included.
Oh, let's think. Sorry, you have a question?
At the end, how are you doing the adhesion?
So there's a Leonard-Jones potential between the cell and the substrate, and this potential is weighted by the surface density of these binder proteins.
Okay, but it's somehow only defined on the surface.
Well, the potential is defined in RD, of course.
Right.
Yeah, but I only need its values on the surface because only the surface moves the substrate.
Okay, so you're integrating on the surface this Leonard-Jones potential.
Yeah, exactly.
Okay.
Yeah.
There is no other call. Let's thank Shikar again.